morning. This is Mizanur Rahman, student, Department of Computer Science and Engineering, Pabna University of Science and Technology. Now, I am presenting a presentation, Machine Learning Based Cancer Subtype Classification. It's for a Bioinformatics course at Pabna University of Science and Technology. The next slide. Cancer is actually highly heterogeneous. That means different patients have very different genomic profiles even for the same cancer types. Traditional one-size-fits-all treatments often fail, making accurate molecular subtyping like driver sub-SBM framework essential for personalized medicine. Then we can discuss about subtyping. Every patient's tumor has unique alterations. Grouping patients into subtypes allow for more targeted and effective therapies because similar subtypes often respond similarly to treatment. Problem statement. Current methods have flaws. Single omics lack depth. Deep learning lacks interpretability, and driver gene methods often ignore individual patients' differences. There is a need for a model that balances individual patients' data with shared coherent pattern. Then we can discuss about driver sub SBM framework. Actually, it is a four-step pipeline: integration, personalization, consensus, classification. Integration, which means building a bipartite graph from multi-omics data. Personalization means ranking genes per patient using random walk and BPR. Consensus means selecting global drivers using the condirect method. Classification means using SBM with SHAP analysis for interpretability. Then we can talk about methodology. Data like somatic mutations and transcriptomic counts are integrated into a network using databases like Signal-Link3 and Reactomes. This captures how mutated genes influence gene expressions within a functional network. Personalizations uses Bayesian personalized ranking (BPR) to distinguish between driver and passenger genes for each specific patient. This step is crucial for capturing the unique heterogeneity that general cohort-level methods often miss. Global consensus. The condirect method identifies winner genes that are important across the whole group. This reduces data complexity while keeping the most biological relevant feature for classifications. Next slide. Experimental data. The model was tested on TCGA dataset for breast, BRCA, thyroid, THCA, and gastric STAD cancers. Actually, it involved over 1,800 labeled samples and thousands of input genes. Classification results. Driver sub SBM achieved high accuracy, which is 94.91% for BRCA, 96.73% for THCA, and 95.95% for STAD. It significantly outperformed existing state-of-the-art models. Removing personalized drivers causes the biggest drop in accuracy, which is minus 20.22%, providing that individual patient data is the most valuable part of the model. Pathway enrichment analysis showed that global driver genes are linked to major cancer pathways like PL3K, AKT, and HPV infections. This shared pathway suggests potential targets for pan-cancer treatments. Biomarker identification using SHAP values, specific biomarkers like PIK3CA, TP53, and GAT3 were identified as key drivers for classifications. Key achievement: peak accuracy of 96.7% and over 485 identified markers for BRCA, successfully processed over 1000 samples and beat four major baseline models. Then discussions and limitations. High accuracy and interpretable results. Limitations: requires pre-labeled samples and cannot yet identify brand new subgroups. Future work includes integrating deep learning and clinical survival outcomes. Finally, conclusion: Driver sub SVM is a novel framework that bridges individual patient biology with population-wide patterns. It advanced precision medicine by providing a more accurate and interpretable way to classify cancer subtypes. Thank you very much. Okay.